All right, so today we're not going to get into physiology today. Today we are really just going to talk about our 3 types of muscle, which you all should already know, right? So you should already know that we've got skeletal, cardiac, and smooth muscle. I'll give you a quick recap on all of those. We're also going to walk through mostly just skeletal muscle today and in this chapter. And we're going to go through sort of the microscopic anatomy of our skeletal muscle. What does it look like? What does an individual muscle cell look like? What are the parts and pieces? So we're going to really get down into that. It's really important that you understand that before we get into the physiology, because we're going to talk about all these microscopic parts and pieces as we do the physiology. Now for physiology, on Wednesday, we'll start into physiology. I do this a little bit differently in that I like to call it chalking and talking. I draw a lot in the physiology part. In fact, I will draw out muscle physiology from start to finish. And I always tell my students, you know, if you can draw out muscle physiology the way I draw it out, then you can answer every question on muscle phys on the exam. So I think there's something about being able to draw it out and see what's happening that's really helpful. So we will do a lot of drawing on Wednesday. So if you attend the live lectures, make sure that you've got a piece of paper, you've got some colored pencils with you so that you can draw out. And we're going to draw it out over and over and over again. So should be pretty helpful. Okay. Now this is a really long chapter, so we're not going to get it done in 1 week. So in fact, we will continue lecturing on this next week as well. So next week, we'll walk through sort of the rest of chapter 9 and we'll talk about how your entire muscles contract. How does that happen? And we'll touch a little bit on smooth muscle. So that being said, I've given you 2 weeks to do this chapter. So the only thing that is due this Sunday would be your chicken quarter dissection for bonus, if you choose to take advantage of that, and your lab exam. Your chapter 9 lab is not due this Sunday. It's due the next Sunday. Okay, you've got 2 weeks for this chapter. So that's helpful. So in terms of our types of muscle and what we're going to walk through today, remember we have 3 types of muscle. We have skeletal, we have cardiac, which we find in the heart, and we have smooth, which we find in our hollow organs, right? So we find smooth muscle in places like our digestive tract. We find smooth muscle in our blood vessels. Obviously, these 3 types of muscle are going to differ in their structure. You know, what do they look like? What is their structure? It's all different for the 3 of these. You know, if we look at these under a microscope, it's really easy to identify cardiac muscle, right? Because it's got intercalated discs, whereas the other 2 don't have that. So their structures are very different. Obviously, where they're found is very different. We find skeletal muscle attached to our skeleton, cardiac muscle in the heart, and smooth muscle in these hollow organs. Also, their function and their means of activation are very different. Only one of these 3 types of muscle is under voluntary control, right? Your brain tells it to contract, and that's skeletal muscle. The other 2 types, your heart, cardiac muscle, and smooth muscle, like your GI tract, these are involuntary. So we're going to walk through a little bit of some of the terminology that we'll use in this chapter. And in fact, you're going to see this a lot throughout the rest of the semester and even in anatomy too. So one thing that we always say is that when we're dealing with skeletal muscle cells, normally if you think of a muscle cell, right, it's sort of, or not a muscle cell, but an animal cell, right? You think of a round cell with a nucleus in the middle. Contains the DNA. Then you've got ribosomes and mitochondria and Golgi apparatus, right? You've got all these different things, these organelles inside the cell. Skeletal muscles are a little different. Skeletal muscle cells are not nice and round. Skeletal muscle cells look more like this, right? I want you all to think about when you look at it under a microscope, remember the cells are really long. They're so long they run off the field of view of a microscope. And so because of that, because the cells aren't nice and round, instead they're really long, we call them muscle fibers. It is the same thing. Muscle fibers. Now, in terms of a muscle cell, we know that our muscle cells are muscle fibers. They contract, and the contraction depends on something called myofilaments. So in order for that muscle fiber, or that cell, to get shorter, you've got to have little myofilaments inside called actin and myosin. We're going to spend a lot of time talking about actin and myosin in a little bit. These myofilaments, the reason why I'm bringing them up now is because I want you to get used to these prefixes. Muscle terminology is always very similar. You'll see prefixes like myo, mice, and sarco. And all of these refer to the muscle, okay? So here's a great example, right? Myofilaments, muscle filaments. The sarcolemma. Sarco also means muscle. So normally, if you look at the outside of a cell, that's called our plasma membrane. In a muscle cell, we call it, instead of a plasma membrane, we call it a sarcolemma. It's the same thing, we just give it a different name. And, inside of a cell, we have that gooey, squishy stuff that all of the organelles float in called cytoplasm, right? Cyto means cell. Well, in a muscle cell, we call it sarcoplasm because sarco means muscle. So a lot of these terms at the beginning, you're going to see myo, mice, or sarco. Okay? So I'm going to walk you through our skeletal muscle and then our cardiac and our smooth muscle. Okay, so let's start with skeletal muscle. Skeletal muscle is the kind that's going to attach to your skeleton, right? Skeletal muscle, that's what's attached to your skeleton. It does have striations, right? Those little stripes that we can see under the microscope. It is under voluntary control. So you consciously tell it to contract. And one thing that's really cool, I think, about skeletal muscle is that it can contract really fast, but unfortunately it also tires really easily. I don't know if the camera will move with me. You can see me better over here. So what a good example of this would be like, y'all remember when you were I remember like in elementary school, we used to have this presidential fitness award. I don't even know if do they still do that? I don't know. But we had to do the standing wall sit, where you have to like get on the wall and like hold that squat position for a while. I actually have an aerobics instructor that will make us do that at the end of every class. We have to do a 2-minute wall sit. And for the first, like 30 seconds, it's no big deal. And then after 30 seconds, your quadriceps start shaking, right? And your legs get really tired and you really want to stand up. And that's a great example of how you've got these big muscles, these skeletal muscles in your body that can contract really quickly and give you lots of power, but they also get tired very easily. And that is very different from a lot of the other muscles in our body, right? Think of your cardiac muscle. Does it get tired? I hope not. That would be bad. Think of how often it's contracting, right? A resting heart rate is around 70 beats per minute. So it's contracting 70 times a minute. And so it's not getting tired. It's going to keep doing that. Same thing with smooth muscle in your body. It's constantly contracting, but it's not getting tired. So skeletal muscle does have a tendency to tire easily. Obviously, this is going to be responsible for your overall body movement. We also say that skeletal muscle is incredibly adaptable, meaning that you have muscles that can exert a huge amount of force, but they can also exert just a fraction of that if needed. So a really good example of that would be like, I can use all of my hand flexors, right? They cause my hand to flex like this. And I can use these to open a jar, right? So I can squeeze really hard and open that jar. I can use the exact same muscles and very gently, when my son gets a splinter, can pull a splinter out. It's the same muscles, but I can exert very different amounts of force depending on what I need, okay? So skeletal muscles are very adaptable. Now, cardiac muscle, again, we only find this in our heart. It is striated, so it does have those little striations, but one big difference compared to skeletal muscle is that it is not voluntary, right? It's involuntary. It is not under conscious control. And it contracts at a very steady rate, and it's actually, the pace of your heart contraction is set by a pacemaker. So you may have heard of a patient having to have a pacemaker put in, and that happens when their natural pacemaker is not setting a good rhythm. Maybe the rhythm is too slow to supply their body and their tissues with oxygen. And so they have to have an artificial pacemaker put in. A lot of times these are some electrical lead wires that get put down into the right atrium of the heart. And then there's a little battery that's sort of implanted in the chest, okay? But you do have a natural pacemaker. Also, your heart is under some neurological controls. So you have nerves that are coming from your brain and from your spinal cord that go to the heart, and they can help your heart respond to your body's needs and demands. So, you know, when you're working out and your CO2 level, which is a waste, starts going up, your body goes, oh gosh, we got to make some changes. One change you're going to make is you're going to start breathing faster, right? Let's get rid of that CO2. The other change you're going to make is your heart is going to start pumping faster. It's going to beat faster to help get oxygen to your muscles faster. So your heart is also under nervous control as well. Now, smooth muscle, this is the type that we find in our hollow visceral organs. Now, I've used that term before in chapter 1. Remember, your viscera are your organs. Okay, so I told you when we went through and we talked about like our serous membranes. Remember, we have a parietal serosa and a visceral serosa. That term visceral just means organ, right? So your hollow visceral organs, that is what your smooth muscle is going to be found inside. This is just going to help to force things through those hollow passageways. It's going to help to move food through your body. It's going to help to move blood. It's going to help to move lymphatic fluid. Now, remember, smooth muscle under a microscope, it is not striated. And this one also obviously is involuntary, right? It is not under voluntary control. You do not have to think about this. Now, the type of contractions we get with smooth muscle are very slow, sustained contractions. Let me come over here because I think I see my tools over here. All right, hold on. Let's see. This board is crazy. All right, I thought maybe I could get it to work. Just kidding. All right, so these are our 3 types of muscles that you can see. We have skeletal, cardiac, and smooth. Okay, so skeletal, you can see attached to the skeleton. Again, this picture should look really familiar to you. That's our skeletal muscle. You can see those muscle fibers, those muscle cells, right? See how long they are? They're so long, they're running off the field of view. We can see in the middle is our cardiac muscle found in the heart. Now, again, this one should be pretty easy to identify, right? We've got these intercalated discs. Those are going to allow the heart muscle cells to contract, get shorter in unison. They're also going to prevent the cells from tearing apart from each other. It holds the cells and anchors them together. And then we also have our smooth muscle, right? This is going to be found in hollow organs like the intestines, the GI tract, the stomach, the lymphatic vessels, blood vessels. And smooth muscle, you can see, does not have any striations in it. So that should have just been a review, right? Y'all should be familiar with our 3 types of muscle. We talked about these when we went through histology. Okay. Now let's talk a little bit about the functional characteristics of our muscle. We always talk about the function of whatever organ system we're discussing for each chapter. So the function of our skeletal muscle, the very first thing is all of your muscles have to be excitable or irritable. So when we're talking about our skeletal muscles, right, they have to have the ability to receive and respond to a stimulus. Now, we've just talked about skeletal muscle, and I told you that skeletal muscle is under voluntary control, meaning your brain tells the muscle when to contract, right? So that means that you have nerves going to all of your skeletal muscle, right? And those nerves are sending an impulse down the nerve, and then from the nerve, it's going into the skeletal muscle. And the skeletal muscle knows, oh, gotta contract. So this is how your brain is telling those muscles when to contract. And the only way this works is if your skeletal muscles are excitable or we call irritable. They have to be able to receive and respond to that nervous impulse. Now, most of the time when we're talking about our skeletal muscles, what's causing them to contract is an impulse is going to come down a nerve. So maybe it's coming from your brain. You're going to say, hey, I want to pick up this pen. So the brain's going to send an impulse down a nerve, out into the muscles. And from there, when that impulse gets to the end of the nerve releases a chemical into the muscle that tells the muscle to contract. And that chemical is called a neurotransmitter. Okay, so we're going to talk a lot about neurotransmitters. A little bit later, we'll talk about these on Wednesday. But neurotransmitters are typically what is causing your muscles to contract. But sometimes your skeletal muscles can also contract based on local pH changes, right? So like a great example, have y'all ever gotten that weird eyelid twitchy thing where your eyelid just like jumps and twitches? That's not your brain telling your eyelid to do that. Nobody in their right mind would do that on purpose, right? That's aggravating. That is typically caused by a local pH change in that tissue. Sometimes it happens when we're dehydrated. Sometimes you might get those twitches more often if you're on a certain medication, but changing the pH can also trigger a muscle contraction. And so this is why we say that excitability or irritability is the ability to receive and respond to a stimulus, usually a neurotransmitter, but sometimes it can be something else, like a pH change, okay? We also say that your muscles have to be able to contract. Those cells have to be able to go from being this really long cell to getting shorter. Has to have the ability to shorten. But on the flip side, it also has to have the ability to be stretched out, okay? So you have to be able to get it shorter and stretch it back out. That's called extensibility. So contractility and extensibility. And then elasticity means that if you stretch that muscle out, it's going to recoil and come back to its original resting length, okay? So a great example would be, let's say that you have just worked out, okay? So maybe you have done biceps, you've done some triceps, and now you've got to stretch those muscles out, okay? So, you know, we stretch our triceps this way, okay, right? You put it over your head and kind of give it a good stretch. So that's extensibility. When I let it go, it has to be able to come back into shape. That's elasticity. So you can stretch it, it's got to come back. Now, we also have some other functions for our muscle. They do perform some important functions for the body. One would be body movement. Now, as we go through these, you're going to tell me which muscle provides these different functions, right? So which one is involved in overall body movement? Y'all have to tell me.
Skeletal.
Yes. So that one's skeletal muscle. All right. What is pumping your blood through your body?
Cardiac.
Cardiac, awesome. And what is allowing substances to move through hollow organs?
Smooth.
Great. Okay. How about posture? Being able to just stand up straight.
Skeletal.
Yes, skeletal muscle. So one thing that I'll mention though about your postural muscles is these are a little bit different. So normally I told you skeletal muscles can contract, but they tire easily. Postural muscles are a little bit different. They're still skeletal muscles, but they do not tire easily. And when I say the word posture, I don't necessarily mean like good posture, shoulders back. I mean being able to hold yourself up against gravity, right? Like I'm standing here and I'm not falling over. I'm holding myself up against gravity and it's not exhausting. Even if, right now, if you're sitting in a chair watching the lecture, you're sitting there and you're not falling over with gravity and you're not exhausted sitting there. You're still using muscles sitting up straight, okay? Now this one we just talked about in chapter 8. It is the most important thing for joint stability. Which one stabilizes your joints? Skeletal muscle. So y'all remember that the tighter, more tone your muscles are, the more stable your joints are. So having nice, tight skeletal muscles means you have nice, stabilized joints. And which one generates body heat?
Spoon.
Yeah, so really all of them will generate heat, but mostly it's going to be skeletal muscle. And you all know this because when you work out, you get hot, you sweat to cool off, right? You're using your muscles more, they're generating more body heat. Did I skip one? No. All right, so we've gone through the function of our muscles. So what I want to walk you through now is we're going to go through the anatomy of our skeletal muscles. Now, again, we're going to use a lot of terms. We're going to look at some different structures in our skeletal muscles. And it is really important that you make sure that you know these different structures before we lecture on muscle physiology on Wednesday. Otherwise, you'll be really lost when we do muscle physiology. Okay, so between now and Wednesday, as we go through all these structures, you need to go back and just study these, make sure you know these terms. So we're going to do gross anatomy first, and then we're going to drill deeper. We're going to do microscopic anatomy of the muscle as well. Now notice that we are just talking right now, and the majority of this lecture is really going to focus mostly on our skeletal muscles, okay? Not cardiac, not smooth. We will focus on cardiac muscle in anatomy 2. So we will talk all about the heart in anatomy 2. And we'll focus a little on smooth muscle at the end of this chapter, okay? But this chapter is mostly on skeletal muscle. So when we talk about the gross anatomy, remember that term means that we can see it with the naked eye. Right, so these are large enough, these are structures that are large enough that we can see them without the use of a microscope. Now your muscles, like your bicep, your tricep, these are discrete organs. How many structures do you have to have for something to be, well, how many tissues do you have to have for something to be considered an organ? At least
2.
2, yes, at least 2. Most of our organs have all 4 types of tissues. Okay, so in our muscles, let's think of the tissues that we have. Obviously, we're going to have skeletal muscle. That's one. Your skeletal muscles are going to have a blood supply feeding them. So they're going to have blood vessels coming into them so that you can get oxygen and nutrients to those cells, those muscle cells. So you've got blood vessels. That's blood. The vessel itself has connective tissue and smooth muscle in it. So we've already got lots of different tissues. Remember, your skeletal muscles, they contract voluntarily. So your brain tells them to contract. So you're going to have nerves going to all of your skeletal muscles. That's another kind of tissue, right? Nervous tissue. Your skeletal muscles are also individual discrete organs. So each skeletal muscle, like your bicep, for example, Your bicep is surrounded with connective tissue. It has this sort of connective tissue covering. And then you're also going to have some attachments in there. Like you'll have tendons as well, right? Which is a different kind of tissue. It's that dense, regular connective tissue. So right there, just kind of rattling off all these different structures, we've got a lot of different tissues in our muscles. So I want to go through some of these different structures with you. So I told you that each muscle, I want you to think in your mind, like think about a whole muscle like a bicep. Your bicep is served by a nerve that's going to come in. So one nerve comes in and then branches profusely. An artery is going to come in and branch profusely so it can hit all the cells. And then typically it's going to drain and be drained. So all the waste, all the CO2, we're going to pull that out through veins. Okay, so we say that the nerve is going to control the contraction, the artery is bringing in the good stuff, and the veins are removing the waste. And all of these are sort of entering into that muscle at a central portion, and then they're going to branch profusely. Now, I also told you that your muscles have this sort of connective tissue sheath around them. There are 3 connective tissue sheaths around your muscle. And it sort of depends on where we are in the muscle. So there's an endomysium, a paramysium, and an epimysium. Okay, now, y'all know that this term right here, mice, means muscle, right? We talked about that. So even if you don't know what each of these sheaths are, you know that it involves a muscle. Now let's look at the prefix. We have endo, peri, and epi. Okay. So I'm going to start at the bottom one, epi. What does epi mean? Think epidermis.
Above.
Yeah, it means above. So right there, we have just figured out that epimysium means it's above the muscle. This is the connective tissue covering that sits on top of the muscle. So it surrounds the entire muscle. So if you've ever cooked, like with chicken breasts, right? So if you get a whole chicken breast, a lot of times what you'll notice is there's this sort of like, when it's raw, there's this kind of like clear sheath that sometimes you can kind of pull off that chicken breast. That's the epimysium. You're actually pulling that off that muscle, okay? Now, perimysium, peri means around. So we're gonna see that prefix a lot. Peri just means that it goes around. So perimysium is gonna surround something called a muscle fascicle. And I'm going to show you a picture of a muscle fascicle in just a second. A muscle fascicle is sort of like a bundle of muscle cells bundled together. It sort of looks like a bundle of twigs. Okay? And then endomysium. Endo means inside. So this is our deepest connective tissue covering. It's deep inside the muscle, and it surrounds each individual muscle fiber. Remember, a muscle fiber is the same thing as a muscle cell. Okay, so now let's look at these coverings, and we're going to look at this muscle on the screen. So we can see here that we have this whole muscle. So around the entire muscle, we have this epimysium. Remember, epi means above or on top. So that's that sort of white covering that you can see out here. Now, if we drill deeper into the muscle, what they've done here is they've pulled out one muscle fascicle. So you can see this is a muscle fascicle. It's this structure right here. Okay, they pulled that out. And I told you a muscle fascicle is a bundle of muscle cells. Kind of looks like a bundle of twigs. And so wrapped around the fascicle is that perimysium. Okay, so that's that white coating around the outside of the fascicle. Now, if we look inside the fascicle and we pull out one of those muscle cells, one muscle fiber, wrapped around the outside of that muscle fiber is the endomysium, right? That's inside. Now, they're just trying to show it to you right here. Okay, this is the same thing. If we just push that muscle cell back into the fascicle, you can see it wrapped around there. Okay. So those are our three connective tissue coatings: epimysium, paramysium, and endomysium. Now, a couple of other things that I want to mention to you about your skeletal muscles is, remember, your skeletal muscles also have connections. They have attachments. Your skeletal muscles connect to your skeleton, and how they do that is through tendons. Okay? A tendon connects a muscle to a bone. So remember, I'll write tendon up here. Whoops. And I'll just write M to B, muscle to bone. Okay. Now remember, we're going to talk about these terms. I mentioned them in the last chapter when we did joints. I'm mentioning them now, and I'm going to mention them again in the next chapter, chapter 10, when we do whole muscles. Remember, your skeletal muscles, where they attach on your skeleton, they're always going to span a joint, right? So like, for example, your bicep is actually going to span the elbow joint, so that when this muscle contracts, you get flexion of the arm, right? And the reason it has to span a joint is because if your bicep just ran from the top of the humerus to the bottom and it tried to contract, nothing would happen. So it has to span a joint to get movement, right? And remember, where your muscle connects at two places, it's the origin or the insertion. We say the origin is always the bone that's not moving when the muscle contracts. The insertion is the movable bone. So again, let's go back to your bicep. Your bicep actually runs from like the coracoid process up here all the way down to the radius and the ulna. And so when your bicep contracts, you get this movement, right? Flexion at the elbow. And so we would say that the origin, the immovable bone, is up here at the coracoid process. The insertion, the movable bone, is down here at the radius and the ulna, okay? And typically we see that when a muscle contracts, the insertion gets closer to the origin. Now, for the types of attachments that we have with our skeletal muscles, what we've been talking about so far, like tendons, those are called indirect attachments. Those are the most common kind of attachment that we can have, right? So these are indirect attachments. It's where you've got connective tissue coming off the muscle connecting to a bone. We call that a tendon. There's also something called an aponeuroses. So a tendon connects a muscle to a bone. An aponeuroses connects a muscle to another muscle, okay? And typically, tendons and aponeuroses, these are way more common because they're small, they're very durable. And so they're really helpful when they're spanning a joint, because what you don't want is you don't want like the belly of the muscle, like your bicep is bigger. I mean, mine's not, but some of you probably have a big bicep, but your bicep is usually bigger right here, right? Well, you don't want this big old muscle crossing through your elbow joint. You'd be like this, right? You'd look funny. So having these small tendons gives you more flexibility, but they're also more durable. Now, the other way your muscles can attach is directly, meaning the muscle basically just connects to the bone. It fuses to the periosteum of the bone. We don't see these often, but we can see these, okay? A couple of examples that we would have of these is some of your cranial muscles do this. Also, a muscle down in the front of your leg. It's that muscle in your shin. And sometimes when you run or you walk, you get shin splints. That muscle also has a very tight, direct attachment to that bone, to the tibia, Okay? And we call that muscle the tibialis anterior. Okay? So it has this really tight connection onto that bone. And that's one of the reasons why you end up with shin splints. When you work a muscle, the muscle actually swells up a little bit. It gets fatter, it gets beefier, right? The way I always remember it is there's some movie where a guy's getting ready to go on a date with a girl and he starts doing push-ups. And he's doing that to make his muscles look a little bit bigger, right? Because when you work them out, they do swell up a little bit. And so when you run or you walk really fast, that muscle in your shin will swell up because it's connected so tight to the bone. It actually has nowhere to go. And it's wrapped really tight in that epimysium, and it actually kind of strangles itself. So it pushes on its own blood supply and its own nerve supply, which is why you get that numbness and tingly feeling. So this is just an overview of our gross anatomy, some of the things that we can see with the naked eye, right? So again, you know, we can see those connective tissue coverings, right? The epimysium. Now, you're looking at a whole muscle at the top, right? Now, if you look at the part at the bottom, what we've done is taken out one fascicle, and that's what you're looking at down here. So here's the fascicle, and remember, around a fascicle is the paramysium. And then here is our one muscle cell, and that is surrounded by the endomysium. Some other things are things like tendons that we can see as well. We can see our nerves, we can see our blood vessels. So now that we've gone through our gross anatomy, let's drill deep into a muscle cell and look at the microscopic anatomy. I know I've mentioned this already, but this is the important part for you to remember so that when we get to muscle phys on Wednesday, you'll know the parts and pieces we're talking about, okay? So each muscle cell, each muscle fiber, remember fiber and cell mean the same thing here. It's very long, and skeletal muscle cells, remember they have multiple nuclei in them, right? When we look at a skeletal muscle cell under a microscope, we see striations, and we see lots of nuclei kind of pushed to the outside. We're going to talk about both of those things in just a minute. Now, in terms of your skeletal muscle cells, those fibers, they are somewhere between 10 to 100 micrometers in diameter. So they're not really wide. They can be up to 100 centimeters in length. So they can be very long. And the reason why you have many nuclei in one single skeletal muscle cell is because in an embryo, how skeletal muscle cells are formed is through the fusion of many embryonic cells. So many cells come together to make one long muscle cell. So that should make sense why you have many nuclei in that one muscle cell. Now, some of the structures that we'll find inside of our muscle cells, now remember, we give these different terms. So one is, obviously, you're going to have a plasma membrane, right? So around the outside of the cell, you're going to have something that holds all of the organelles in place. And we call a plasma membrane of a muscle the sarcolemma. There's also going to be cytoplasm inside your muscle cell, but again, we call it sarcoplasm. And the reason why we give it a different name is because sarcoplasm is different than cytoplasm. It is specialized for the muscle. It has two things in it that regular cytoplasm would not have. One is sarcoplasm has a lot of glycosomes. Glycosomes are little vesicles that store glycogen. Does anybody know what glycogen is? Like what kind of macromolecule it is? It's basically a sugar or a carbohydrate. Okay. The other thing that sarcoplasm will have in it is an oxygen binding protein called myoglobin. Right? That sounds real similar to hemoglobin. Hemoglobin is an oxygen binding protein found in your blood. Heme means blood. Well, myoglobin is an oxygen binding protein found in your muscle because myo means muscle. So now we've got, in our sarcoplasm, we've got two things. One is storing carbs and one is storing oxygen. There is a reason for this, and it is cellular respiration. So I like to write cell respiration. I like to write the equation this way. I write it very simply so you can just sort of remember. And I think in anatomy, it's really helpful to write it this way so that you can really remember these molecules. So carbs plus oxygen, so carbohydrates plus oxygen form CO2, which is a waste, right? We breathe this out, water, and ATP, right? That is cellular respiration. Now, in reality, you've probably learned cell respiration as glucose plus oxygen forms CO2, water, and ATP. I like to say carbs because glucose is a sugar, carbs are sugars. And it's easier in your mind if you think about carbohydrates because now you start thinking about food stuff. So you've got to eat and consume carbohydrates, and you've got to be breathing in oxygen so that you can make energy, ATP. The whole point of cell respiration is making energy. And in your muscle cells, you have to have energy. You have to have ATP for them to be able to contract. In fact, your body uses your own body weight in ATP every day. So you are going to essentially produce your body weight in ATP every day, but you burn it as fast as you make it. So your muscles have a very heavy dependence on ATP. They need ATP. So it makes sense to me why now in your sarcoplasm, you have little vesicles that store carbs and little vesicles that store oxygen. You've got the two things right there that you need to make ATP, okay? Now, your muscle cells are also going to have some other things. Inside your muscle cells, you are going to have myofibrils, right? These are things like actin and myosin. So we're going to spend quite a bit of time talking about these. These help your muscle contract. Without these, you can't get a contraction. You also have some other things. You have a sarcoplasmic reticulum. That should sound really familiar to you, right? A sarcoplasmic reticulum is basically what we find in our muscle cells, the equivalent of what we find in our body cells. And then we also have something called T-tubules, and I'll walk you through those, okay? So let's talk about our myofibrils first. And I'm going to orient you in terms of what you're looking at in this picture. So again, think of an animal cell, right? A round cell with a nucleus in the middle that has our DNA. And then it has vesicles, ribosomes, mitochondria, Golgi apparatus, all those structures, right? Muscle cells we already know are different, right? They're very long. We call them muscle fibers. So this is a muscle cell. Now I want you to look inside that muscle cell. Inside that muscle cell, you see all these little rods, right? Those are myofibrils. So the inside of your muscle cell is densely packed with these myofibrils. So I'm going to go back to this page. Remember, I just told you that myofibrils are what cause and help your muscle to contract. Makes sense that the entire inside of your muscle cell is just packed with myofibrils because that's the job of your muscle, is to contract. Now, you're still going to have all those organelles that we talked about. You still have nuclei. You see the nuclei right here? Remember, we have multiple nuclei in skeletal muscle. You still have mitochondria. You still have ribosomes. You still have all those structures. But what happens is they kind of squeeze in where they fit. So remember how I told you that skeletal muscle has striations? We're going to talk about those in a minute. but it has these multiple peripheral nuclei, so the nuclei are kind of mushed to the outside. This is why. Because the inside of that muscle cell is so packed with myofibrils that the nuclei end up squished towards the outside, okay? So these myofibrils are making up most of the volume inside of that cell. All of the other organelles are just sort of squeezed in where they fit. So now what I want to do is I'm going to walk you through these myofibrils. So we're just going to keep drilling down and looking at these tiny, tiny things. So we know these myofibrils play a huge role in getting your muscle cell to contract. So let's look at what is inside of a myofibril. So all we've done in this picture is just taken one myofibril. I'm going to go back. And we're just going to take this one myofibril and blow it up so you can see it. Okay, so here's our myofibril. Inside our myofibrils, they're composed of actin and myosin. A lot of times you might hear myosin as described as the thick filament because it looks thicker. And actin, a lot of times, is described as the thin filament. It just looks skinnier. Now, if we look at this myofibril at the bottom, you should kind of be able to see here which of these, the red one or the blue one, looks thicker. The red one is a lot thicker. So that's our myosin, that's our thick filament. And those blue lines in there, those are actin. Those are our thin filaments. So before we talk about myosin and actin, let's look at this arrangement of actin and myosin. So before we really look at the individual molecules, let's look at how they're arranged, right? So we have our thick and our thin filaments. And obviously there is a pattern here within this myofibril as to how actin and myosin are arranged. And we call that pattern, we call the smallest part of it a sarcomere. So a sarcomere runs from Z disc to Z disc. Now, if you look at this picture, if you didn't know what a Z disc was, you should be able to look at that picture and go, oh, that's right here, right? It's the part right here that looks like a zigzag, right? That's a Z disc and that's a Z disc, right? So a sarcomere basically runs from Z disc to Z disc. We say that a sarcomere is the smallest contractile unit of a muscle cell. If you get smaller than a sarcomere, you don't get a contraction. You have to have this unit to see a contraction. And I'll show you a model of a sarcomere in just a minute. So those Z discs are basically proteins that are anchoring those thin filaments together. Now you can tell because notice that it's all blue. So that's our actin, our thin filaments. And it's only touching those thin actin filaments. It's not touching the thick myosin ones that are in red. So Z discs are just anchoring those thin filaments together. A couple of other structures that we see within this sort of pattern. We have what are called A bands and I bands. So an A band is from here to here. Okay, so you can see us right here. Here's our A band. And I remember this. I remember A because I think of the word dark, and dark has an A in it. And I remember that because if you think about it, this is an area in this sarcomere that has both thick and thin filaments in it. So under a microscope, because both of these filaments are found, it's going to have a darker appearance to it. The I bands are these regions, here and here, okay? Circle this one so it's easier to see, and I'll circle that one. All right, so there's our I bands. Notice you don't have any red, thick filaments there, only our thin actin filaments. So I remember that I band, and I think of the word light. And light has an I in it. So I remember, okay, the I band is going to look light under a microscope because it doesn't have any thick filaments. It's all just thin filaments. So if we were to look at these just under a microscope, we could see that the A band looks darker because everything's in there. The I band looks lighter because it's only our thin filaments. A couple other things that we see is something called an H zone. So you can see the H zone right here. And the H zone is just this region right here. I'll draw a line down one side and then the other. Circling it. Okay, and notice that in the H zone, so this is in the middle of the A band. Notice it's only our thick filaments in there. Okay. And then lastly, we have these little M lines. And the M lines, you can see them right here. There are these lines right here. You can see them, they've labeled them over here. Here's our M lines. And the M lines are the proteins that are holding those thick filaments together. So remember, Z discs hold thin filaments together. M lines hold thick filaments together. Now, here's a picture looking at a myofibril under the microscope. And what we can see is we can see the arrangement within that sarcomere. We can see the A bands and the I bands, right? So if we look, I know this one looks a little different, but it is a sarcomere. So we have our Z discs here, right? A sarcomere is from Z disc to Z disc. And remember, right in the middle, we have our A band, both thick and thin filaments. Look at it up here under the scope. Looks dark. Remember A for dark? Over here, we have our I band, right? Only gonna find those actin, that thin filament. So up here, notice it looks light in color. So remember, I for light. So I told you towards the beginning of this lecture that I would tell you why in a skeletal muscle cell, the nuclei are always pushed to the outside, right? That's because inside that muscle cell, you're filled up with these myofibrils. So the nuclei get squeezed to the outside. I also told you why you find those striations. Now you know why muscle cells under a microscope appear striated. Because of the arrangement of actin and myosin. So if we put all these myofibrils together, and if you look at this myofibril here, you can actually see those arrangements of sarcomeres in there. And if we put them all together, those sarcomeres line up, and you can see we've got our A band where it's dark, right? And then we've got these I bands where it's light in color. And you can see the arrangement is just A band, I band, A band, I band. And it gives you a striated appearance. Okay. So now that we kind of know how our thick filaments and thin filaments are sort of arranged to create a sarcomere, what I want to do is drill down and look at our thick and thin filaments. Let's talk about actin and myosin. Okay. So myosin is our thick filament. It's the one that's in red. And to me, myosin molecule, so one myosin molecule, it almost looks like if you have two big golf clubs, like driver clubs, the big ones, and you were to hold them together and then wrap the handles around each other. That's what a myosin molecule looks like. So here's a picture. This is a myosin molecule at the bottom, right? It kind of looks like that. Kind of looks like two big golf clubs with the handles wrapped around each other. So each of your myosin molecules has a tail, that'd be like the handle of a golf club, and it has two heads on it, two big globular heads. Now, in order to make a thick filament, we need about 300 of these molecules. And that will create a thick filament. So a thick filament made of myosin has about 300 myosin molecules in it. And you can see all of these myosin heads scattered all over the place. Now let's look at actin. Let's look at our thin filaments, right? So thin filaments are the blue ones made of actin. And to me, actin looks very different from myosin. Actin looks to me like little beads. I don't even know if they have these anymore. When I was growing up, they had pop beads where you could like stick them together so they had little holes in them. And so these almost remind me of those, right? So they have these sort of divots cut out. You'll see those divots in there. So it really looks like little beads. And a thin filament almost looks like a pearl necklace that's just sort of been twisted. So you can see all those beads strung together here in that thin filament. So that's our actin, makes up that thin filament. Now I want to come back and talk to you a little bit about what those divots are. So those divots on those actin molecules are called active sites. And the active sites are actually where the myosin head is going to latch on to. So myosin and actin will actually connect to each other. And myosin will connect onto actin at that active site. Okay, so it's going to connect at those little divots. Now you'll notice up here when we look at our thin filament, notice that you've got this sort of brown and yellow structures right here, right? Here's the brown. And notice what it's covering. It is directly on top of that active site. Those are regulatory proteins called troponin and tropomyosin. So you can see tropomyosin is that brown string, and troponin helps to hold that in place. Okay. So here's a picture showing you actin and myosin. And when a muscle contracts, what's going to happen is the myosin heads, those little golf club heads, are going to reach up and grab onto actin at the active site, right? So they're going to grab on. You're going to find that active site and grab on. I'm just going to do that one side right there. Once they've grabbed on, they're going to push that actin forward, right? So they're going to push actin in this direction. And that sarcomere is going to get shorter. And that's going to give you a muscle contraction. What you don't want is a muscle contraction all the time. So you only want a muscle contraction when you need one. So I'm coming back to actin and this thin filament so I can show you this. So I told you that actin has these regulatory proteins, troponin and tropomyosin. They cover over the active site. So in my mind, the way I think about it is I think about troponin and tropomyosin is kind of like the chastity belt for your thin filament. So when you don't want a muscle contraction, the chastity belt is on and it's locked, and myosin cannot connect. But when you're ready for a contraction, these regulatory proteins actually shift and they move out of the way. Now the active site's open, myosin heads can latch on, and you can get a contraction. Okay? So these regulatory proteins just prevent you from having undesired contractions. So this is just an image that's kind of walking you through what we've talked about so far, right? So again, right here at the very top, we've just got our muscle cell, our muscle fiber. Remember, inside the cell, we have all these little myofibrils that contain actin and myosin, thick and thin filaments. These are doing the job. These are helping the muscle contract. If we blow up one myofibril and look at it, we can see the arrangement of our thick and thin filaments. Right? And we can see how they're arranged. And remember, we do have a sarcomere as the smallest contractile unit in our muscle. If we just look at the sarcomere by itself, so we just blow that up, we can see that arrangement even better, right? So we can see things like our A band, our I band, our Z disc, our M lines. We can see all those parts of a sarcomere. And then we can also blow up and look even deeper and look at our thick and our thin filaments. We can look at the actin and the myosin. So this is a great image because it just kind of shows you step by step drilling down into that muscle cell. Now the last thing I'm going to walk you through is the two more parts in the microscopic anatomy, the sarcoplasmic reticulum and the T-tubules. This picture is a little bit weird. So we're going to start over here, okay? You are looking at a muscle cell, right? Because here's our muscle cell, it's just in gray. And they're trying to show you right here, they've just pulled out a couple of myofibrils from that muscle cell. And they've blown them up. So now you can see a couple of those myofibrils, right? You can see everything that we've just talked about, A bands, I bands, Z discs. You can see the arrangement in there. But a couple of other things you can see. One, notice that thing that looks like a blue loose knit sweater. Okay? That is called the sarcoplasmic reticulum. And the sarcoplasmic reticulum, if you'll notice, kind of running down the sides of it, this gray structure, that is called the T-tubules. Now, this picture does a decent job trying to show you this, but the T-tubules, those gray structures, those are actually part of the sarcolemma. See that sarcolemma? See that? This runs around the outside of that. Oops. I need to do that. Sorry. Runs around the outside of that cell. You can see that here's the sarcolemma, and notice right here, this is an invagination of the sarcolemma. So we call that the T-tubule. Now that loose knit sweater, that sarcoplasmic reticulum, right next to that T-tubule, it gets really thick, right? So you see on both sides of that T-tubule, that blue loose knit sweater is getting really thick. And we call that the terminal cisternae. So let's talk about those structures. So again, the sarcoplasmic reticulum looks like this sort of loose knit blue sweater that sits on top of all those little myofibrils. And the terminal cisternae is sort of this expanded end right next to the T-tubule. Now the terminal cisternae, their job is to store calcium. Do you remember how we have talked about calcium this semester and how important calcium is? And I told you that blood calcium is so important. You have to maintain a blood calcium level because calcium is used in almost every physiological function. It helps your glands secrete, it helps your cells divide, it helps your nerves send signals, it helps your muscles contract. So calcium is actually the go signal for a muscle contraction. So inside a muscle cell, you have a structure called the terminal cisternae that's storing calcium. So when it's time for a contraction, guess what happens? The terminal cisternae squeeze out a ton of calcium so that you can get a contraction. And then I also showed you those T-tubules. Remember, your T-tubules are basically portions of that sarcolemma, and these penetrate all the way into that muscle cell. And they'll help to conduct nervous impulses all the way into that cell so that the terminal cisternae know when to release calcium. So what I mean by that is, let's say your brain's telling your muscle to contract. Here's an impulse coming down a nerve into your muscle. It's going to jump from the nerve to the muscle. It's going to spread down the sarcolemma, down the T-tubes, and it's going to tell those terminal cisternae to squeeze out calcium. That's the go signal for a contraction, okay? So calcium is that go signal. We say that the T-tubule with the terminal cisternae on either side is called a triad. We actually give it a special name, triad. All right, so I'm going to pull up, the very last thing I'm going to show you is I'm going to pull up a whiteboard really quickly. And I'm just going to use some of the terms. I'm just going to use some of the terms that we've been using. And I'm not walking you through muscle physiology, but I'm just going to prep you for what we're going to talk about on Wednesday. Okay, so just to talk about what we've learned, we know that our skeletal muscles are controlled voluntarily, right? So right here, I've just drawn a neuron. And that neuron is going to come into a muscle cell. Here is my big muscle cell. I'll write down here muscle cell or muscle fiber. Means the same thing, right? And in my muscle cell, remember I have T-tubules. These are part of the sarcolemma that just dig all the way through that muscle cell. So here's a T-tubule. And we have that sarcoplasmic reticulum. I'm not going to draw that all over here, that blue loose knit sweater, because you won't see what's underneath. So instead, I'm just going to draw the expanded ends. Remember, we call those big ends of our sarcoplasmic reticulum, we call those the terminal cisternae. And remember, together, our T-tubules and our terminal cisternae, so all of these are called triads. Now, inside of our muscle cells, remember, inside these muscle fibers, we also have these myofibrils that contain actin and myosin. I'm not going to draw out a zillion myofibrils and a zillion sarcomeres, but I will draw out one sarcomere, okay? So let's do one sarcomere. One sarcomere is going to run from Z disc to Z disc, okay? We've got our thin filaments. And then we have our thick filaments. So those are some of the structures that we've looked at. So what we're going to talk about on Wednesday is how we get a contraction, what's actually happening, right? In reality, when your brain tells your muscle to contract, an impulse is going to come down a neuron. It's going to jump from the neuron into the muscle cell, and it's going to spread down the muscle cell. It's going to spread down the T-tube, and that is going to cause this sarcomere to go from looking like this to looking more like this. It's going to get shorter. Those thin filaments get pushed towards the middle. The Z discs get pushed towards the middle. And the sarcomere gets shorter. All those sarcomeres inside that muscle cell shorten up, the whole muscle cell is going to get shorter. So when we talk about muscle physiology on Wednesday, we're going to kind of talk about it and break it down into 3 parts. So we're going to talk about how does an impulse jump from a neuron into a muscle? How does that happen? Then we're going to talk about how does it spread down the muscle cell and down the T-tubes? How does that happen? And then we're going to talk about how does that sarcomere get shorter? What's happening there? So when we do muscle physiology, we're going to kind of hit those 3 different parts. We're going to break it up into 3 parts, okay? I'm going to grab a sarcomere model. There we go. All right, so y'all should be able to see my sarcomere model here. So this is just a model of a sarcomere, and you can kind of see there's this sort of overlay on it, where you can see in yellow, these are my T-tubules. I have this sort of green loose knit structure, right? That's my sarcoplasmic reticulum, where it's next to the T-tubules. That's my terminal cisternae. Okay, so we can see that. And then I'm going to take this off so that you can see the sarcomere. Now, unfortunately, this model, the colors are backwards from what's in your textbook. So the colors are a little off here, but you can see here on the ends, these are my Z discs, okay? And here are my thin filaments, right? You can see these actin molecules. Let me zoom in just a smidge. Too much. Okay. So you can see these thin filaments right here. These actin looks like little beads. You can see the regulatory proteins, troponin and tropomyosin. And then underneath here, you can see these thick filaments in here. You can see those golf club looking structures, right? Those are myosin heads. So when a muscle contracts, this sarcomere has to get shorter. Now remember, we talked about all the different pieces of a sarcomere, right? We've got our Z discs, we have our thin filaments, we have our thick filaments. I also told you that right here in the middle, where we have thin and thick filaments, that's our A band. It's darker in color. And over here, where you only have thin filaments on each side, that's our I band. It's lighter in color, right? Now we also have our M lines right in the middle, okay? And we have this zone right here in the middle called the H zone, where all we have are these thick filaments. So what's gonna happen when a muscle contracts and the sarcomere gets shorter is the myosin heads latch onto actin and they push actin towards this middle. They push it into this H zone here. They push it towards the M line. And so you can see here, I'm just gonna move this in. So you can see those Z discs are coming together. And you can see that those actin filaments, those thin filaments have gotten pushed towards the middle, okay? We have essentially lost that H zone. It's essentially gone, right? Here was the H zone. We've just shortened that sarcomere, pushed all these thin filaments into that H zone. We've also lost, notice, our I bands, right? At the ends. When we push this together, those are gone. So that sarcomere gets shorter, okay? If all those sarcomeres get shorter, then the whole muscle is gonna contract. And then when the muscle relaxes, the sarcomere lengthens back out again and you can do it all over again. So this is a good model just for the visual.